Okay, good morning. Welcome back. I just a few minutes ago made an announcement to the forum site about the quiz and basically a reason why we're not able to release the quiz results quite just yet, or at least the answers. So just bear with us as we try to accommodate the students who have requested accommodation. So just take note of that for the future quizzes. I laid some rules out, sort of a policy we're going to apply going forward. So just so everyone's aware of how that's going to work. Okay, so that's posted to the forum. You can check that out. Today we're going to have the thrilling conclusion of topic 7, which is carbohydrate structure. And I haven't talked a lot about functions of carbohydrates yet, but this is one conceivable function of carbohydrates as a form of birth control. Here, as Calvin has discovered, that's sort of tongue-in-cheek. Carbohydrates are not actually used for birth control. So where I left off was starting to talk about Haworth projections, and just to refresh your memory, monosaccharides can cyclize, and we represent that in this Haworth projection form, which looks like a planar group of molecules or atoms, but it's not actually planar, and that's what this little diagram on the right is supposed to show. It's got a slightly non-planar configuration. But one of the main points that I tried to make was to get you to see how the relationship, basically see the relationship between the chiral sites, the chiral carbons here, and the positions of these OHs in the Fisher projection versus the Haworth projection. And you should be able to correlate those. Now, it caused some confusion with this carbon 1 here. And in fact, confession, biochemists don't normally draw cyclic carbohydrates in this way. So I just stuck the OH there, and I wasn't really thinking about how it's going to correlate with the OH on the right in the Haworth projection. I was just trying to make this point that this is an awkward way to draw the cyclic structure as a segue to get into the Haworth projections. So the fact is, though, that on this carbon 1 in glucose, remember, it could have an aldehyde form, C double bond O in the linear form, and now it has adopted, it's got an OH group on it. That's the anomeric carbon, okay? And that's the general rule. Anytime you look at a Haworth projection like this, it can be helpful to identify this carbon, the carbon that used to have the carbonyl on it. And the way you can identify that is to look for the carbon that has 2 bonds to oxygen, because in the linear form of the monosaccharide, that carbon had 2 bonds to oxygen, right? A double bond between carbon and oxygen. In this structure here, you can see that same carbon also has 2 bonds to oxygen. It's just 2 different oxygens, okay? Whereas all the other carbons have only 1 bond to oxygen. So if you're ever looking for this anomeric carbon that used to have the carbonyl on it, you just look for the carbon that has 2 bonds to oxygen. And it turns out that the OH on this anomeric carbon can be either above or below the plane of the ring, depending on the angle of attack of the nucleophilic attack that makes the new carbon-oxygen bond. And we need a way to distinguish between these 2 structures. So alpha glucose, the alpha form is the form where the OH on the anomeric carbon is on the opposite side of the ring as the highest numbered carbon in the sugar. And that's carbon 6 up here. So those are opposite sides, that's alpha. And if the OH on that anomeric carbon is on the same side as that highest numbered carbon, then we call that beta. And just to reiterate, beta and same start with consonants, and alpha and opposite start with vowels, and that's an easy way, perhaps, to remember. And some people called me on this. You've shown this as an equilibrium. It's true that these will interchange in between each other, but they're not actually equally favored. So in fact, it happens that the beta glucose form is more favored in solution, but I don't really care that you know that. It's not really the point, okay? So, all right, so that's the anomeric carbon. And just then to try to show a different example, to try to get these concepts firm in your mind, this is another hexose, fructose, and this is actually a ketose, right? The The double bonded oxygen is on carbon 2 in this molecule. So that's a ketose. It also forms a ring structure in which the oxygen on carbon 5 does this nucleophilic attack thing. And then it makes a new carbon-oxygen bond between this oxygen and carbon 2. And so you can see that in this structure here, where I've numbered the carbons, just so you can see. And remember, you start numbering at the end that's closest to the anomeric carbon, or this C double bond O. So we don't call that carbon 1, we call the end closest to that carbon 1. So that's this. You can see the relationship, the numbering, and hopefully you can verify that I have drawn the Haworth projections properly. And particularly, you want to look at carbons 3 and 4 here that are chiral, and you can verify that I've put the OHs in the proper configuration. So, but the point here also is we have two versions of the cyclized fructose molecule. One's alpha fructose, one's beta fructose. And how do you tell the difference? You look at the OH on that anomeric carbon. Question. From the Fisher projection, absolutely not, because the molecule itself doesn't know whether it's going to be alpha or beta until the cyclization happens. Okay, so and as you can see, I've drawn them as equilibria. And so the cyclic ring is going to close and then open and then close and then open and close and open, a gazillion times while this molecule is still existing. And so it's going to interconvert between all these three different forms. Okay. So no, you can't just look at the Fisher projection and say it's going to be alpha or beta. It could be alpha, it could be beta. It all depends on the circumstances of the ring closing. Okay, but you should be able to look at a Haworth projection and tell me, is that an alpha sugar or a beta sugar? And the way you do that, again, just to reiterate, you look at the position of the OH on the anomeric carbon. And wait a minute, how do we tell which one's the anomeric carbon? It's the carbon that has 2 bonds to oxygen. So that'd be carbon 2. So even if you didn't see this Fisher projection, you would know, you should be able to identify that carbon 2 is the anomeric carbon because it has 2 bonds to oxygen. And you look at the OH on that anomeric carbon and you compare its position to the position of the highest numbered carbon. And on the left here, this is opposite in position. So that's alpha fructose. And over here on the right, it's on the same side of this so-called planar ring. And that's beta fructose. Okay. Does that work? Okay, seeing some nods and seeing thumbs up. Okay, so we'll see as we proceed whether that's in your minds or not. But now we're going to get even more complicated because you can take monosaccharides and you can join them together into a disaccharide. And of course, mono means 1, di means 2, tri means 3. It's just a little like polypeptides. The tripeptide has 3 amino acids. A trisaccharide would have 3 sugars. But here we're dealing with the disaccharide. And I'm using as an example lactose, which is found in milk. And you join then beta-galactose and glucose. And I've shown the structures of those here. And you join them at specific place. And it happens that the anomeric carbon here of beta-galactose, and you can tell it's beta, just another aside. And you may feel like I'm beating a dead horse here. But it's beta because the OH on this anomeric carbon is on the same side of the ring as the highest numbered carbon. So that's beta-galactose. And that OH there will undergo a reaction here. Actually, this OH will do an attack on the carbon, but we're not going to get into the mechanism. The basic point is you're losing water from these 2 molecules, and you're making a new carbon-oxygen bond, or basically you're joining the 2 monosaccharides together. So you lose the water. And now you've got this linkage here between the 2, and they're joined together. And this is what we then call lactose. Or if you want to be a little more precise, you could call it beta-galactose 1,4-glucose. And the beta-galactose just indicates that the galactose is in the beta configuration. So even though now it's formed this bond to another molecule of glucose, you can still see that the oxygen is on the same side of the Haworth projection ring. And so we would still say that this is a beta configuration of this sugar on the left. Okay. 1,4, because that indicates the numbers of the carbons that are involved in this bond, right? So it's carbon 1 of galactose. And again, you number starting at the end that's closest to the anomeric carbon. And you might think, well, wait a minute, how do you know what the end is, right? Well, it's a cycle. Where's the end? Well, the end is where you've got a carbon only joined to one other carbon, right? So really, I mean, that's the obvious end right there. The carbon 6, it's just the end. But the anomeric carbon in each of these sugars is only joined to one other carbon, and that would be the end in the linear version, right? So look for that carbon. So we start with number 1 here, and we number that around 2, 3, 4, 5, and 6. And likewise, we can number that around in this molecule. 1 is there, that's the end, that's closest to the anomeric carbon. 2, 3, 4. So it's carbon 4 here that's joined to carbon 1, so we call that a 1,4 linkage. Okay, now this bond here, we call it an O-glycosidic bond because it's a sugar bonded to something else via an oxygen. Another common type is an N-glycosidic bond, and you see that in nucleosides, but we'll get to that in topic 14. So that's an O-glycosidic bond. Now, the interesting thing is that once this bond has formed, the chemistry of the group changes such that the galactose ring here is no longer able to open and close. Okay, so this bond, when it forms, that freezes this structure in the cyclic form. So it's no longer in that equilibrium with the linear form. And so that means that the galactose is fixed in the beta position, the beta configuration. All right. So anytime you see lactose, the galactose is going to be in the beta configuration. And again, I'll just reiterate, you not have to memorize individual sugars here, right? So I'm not going to ask you what is lactose on the exam. I'm just giving you an example. It's pretty common. Commonly used. Okay, now that one is still beta, right? And I'm just emphasizing it's on the same side of the ring, and we still consider that beta. So this you would consider a beta 1,4 linkage. You could just abbreviate it that way. You're asking what kind of linkage is this? It's a beta 1,4 linkage because the anomeric carbon's in the beta position and it's joining carbons 1 and 4. Now this end over here on the glucose side, it is still, that anomeric carbon is still able to undergo this reversible reaction. So it can open or close, open and close. So on the right-hand side of this molecule as drawn, this OH could be in either the beta or the alpha position. I'm showing it in the beta position here, but I could have easily shown it in the alpha position, and that also would have been correct for lactose. So while the one anomeric carbon is frozen because of its O-glycosidic bond, it's always beta in lactose. The other one can open and close. And so then, yeah, so I've drawn it here. As I say, it's this beta because they're on the same side, but I could have drawn it the other way, and that's here. So you would call this structure on the top alpha lactose based on the configuration of this part of the molecule, and we call the thing on the bottom beta lactose, but in each case you have a beta 1,4 linkage joining the two sugars together. Okay, so just because it's alpha lactose, that doesn't mean the linkage between the two sugars is alpha. That's not referring to that. It's referring to the position of the OH over here on the glucose part of the molecule. Okay, sometimes students get a little mixed up about that, so that you have to draw the distinction between the two anomeric carbons. One is behaving this way, it's fixed as beta, the other goes back and forth. Okay? And that's just one example. You can imagine there's tons of examples you can imagine of different combinations of sugars joining together. But in each case, anytime a sugar joins to another one, at least one of the anomeric carbons is involved, because that's where the chemistry happens at the anomeric carbons. So you would never see, you know, say carbons 2 and 2 of this being joined together, because that's just not the way the chemistry works. You have to have an anomeric carbon involved. Yeah.
Let's say the galactose is alpha form.
Yeah, so the question is, if galactose were in the alpha configuration, you can easily imagine that. If we go back, you can imagine drawing a galactose here with the OH on the bottom of the ring in the alpha configuration. Yes, that could in principle undergo a reaction with glucose to make an alpha 1,4 linkage. But we wouldn't call that lactose. That would be some other sugar, okay? Because lactose is defined by beta galactose joining with glucose. And again, you're not responsible for those those sorts of names like that. You should know the system of why this is named this way. So if I show you any sugar, you should be able to tell me, is it a beta or an alpha linkage, or is it a 1,3, 1,4, 1,6 linkage? What is it? Yeah. Does the glycosidic bond only ever happen between carbon 1 and 4? No. We'll see examples coming up where that's not the case. Okay. So here's another one. Again, I can feel like, oh man, how many of these are going to show? But here, this is the one we're going to use to see if you're absorbing all the good wisdom that I'm putting out. Okay, so this is another disaccharide. Glucose and fructose join together to make sucrose. And sucrose is a disaccharide, and this is its structure. So now we'll see if you were listening or if I was making sense or one or both of the two. This will be a target question. So each of these parts has a anomeric carbon. Click on the two anomeric carbons. Yeah, I'll give you a little bit more time. We've got some answers pouring in. All right, let's see what we got. Okay. We have some divergence of opinion, shall we say. Okay, so more carbons than I expected got attention here. We got this one on the right, we got that one there, seems to have gotten a lot, this one here, and various other ones, okay? So now, what's the rule? I gave a rule just a few minutes ago. How do you find the anomeric carbon? It's the carbon that has 2 bonds to oxygen, right? So in each case, the anomeric carbon will have 2 bonds to oxygen. So if you look at these monosaccharides individually and you follow them through, that one only has 1 bond to oxygen. That's only 1 bond to oxygen. That's only 1. That's only 1. That's only 1. But this one has 2 bonds to oxygen. Okay, so that is the carbon that used to be a carbonyl. It used to have 2 bonds to the same oxygen. Now it's been attacked by a separate oxygen. Now it's got 2 bonds to oxygen still, but they're 2 different oxygens. Okay, so on the left-hand side, this one here that I'm pointing to is the anomeric carbon. It has 2 bonds to oxygen. Now on the right side, a lot of people did go for this one over here because generally speaking, in the Haworth projection form, we put the anomeric carbon on the right. That is the convention. But in a molecule like this, I've had to change that because you actually have the 2 anomeric carbons joined together. And it would look really awkward to put the anomeric carbon over here and then try to make a bond with this anomeric carbon. So if you look at this carbon here, you can see that it has 2 bonds to oxygen, according to the rule I said. And that, therefore, is the anomeric carbon there. This one has only 1 bond to oxygen, that one right there. So this is actually not the anomeric carbon, okay? Yeah.
So if both anomeric carbons are in the glycosidic bond,
How do you call it? Have some patience, my friend. We're getting there. Okay. So now, for the next question, I want you not to talk to anybody else. Okay. So you have to answer this next question on your own. No discussion. So I told you where the anomeric carbons are. Now, for this one on the right, simple 50-50 choice. Is this alpha or beta? Go. No talking. The answers come in faster when I don't let you talk. Okay, we'll go to 1 minute. Okay, 1 minute. Okay, beautiful, beautiful. Okay. So we're going to do this again, but now I want you to talk because there's obviously a lot of disagreement in the room. Now we're all friends, remember, we're all friends, so we can disagree and still like each other. But I want you now to talk to your neighbor and convince your neighbor that you are correct. Okay, I'll give you another minute to talk and put your answers in. Okay, 10 more seconds about. Have fewer answers this time. All right. Okay, so we've seen some movement towards beta. And that's a little encouraging, but still 30% of people think it's alpha. So what's the rule? I said to determine alpha or beta, I said you look at the oxygen that's bound to the anomeric carbon. Is it sticking up or down relative to the highest numbered carbon? So we just identified, and I explained in the last question, that this is the anomeric carbon, right? It's the one with 2 bonds of oxygen. So we want to look at this oxygen here, which is the one, sticking below the plane of the ring, and we want to compare the position of that one to the highest numbered carbon. And of course, how do you number the carbons? You start with the end that's closest to the anomeric carbon. So this would be carbon 1, 2, 3, 4, 5, 6. And you see that this oxygen is below the ring, just the same as carbon number 6, the highest numbered carbon. So beta would be the correct answer. All right. Anybody Want to talk about that, or are we good with that? Just the position of the oxygen relative to the highest numbered carbon. Okay, so sucrose is what you get. Oh yeah, here's a question. The anomeric carbon is carbon 2, absolutely. And consistent with everything I've told you throughout this topic, in fructose, the anomeric carbon is carbon 2. That's the one that has 2 bonds to oxygen in it, in the linear form and in the cyclic form. Okay, so it's the carbonyl carbon, carbon 2. So there I've got it numbered. So in sucrose, you take alpha fructose, or sorry, alpha glucose and join it to beta fructose, and you end up getting this structure here. So that is the structure of sucrose. And you'll see that both anomeric carbons are joined in the glycosidic bonds. So neither one of them can open and close. So there's only one structure of sucrose. Does not make any sense to say alpha sucrose or beta sucrose because those two forms don't exist. There's only sucrose. And it has this sort of linkage. Now, as someone said, how would we name this linkage? I would call it this way. You'd say alpha glucose is joined in a 1,2 bond to beta fructose. Or if you want to do a shorthand, you can call it an alpha 1 beta 2 linkage if you really wanted to. Okay. So that's a little tricky because what I've had to do to make the fructose fit here in a nice little structure, I had to basically flip the structure 180 degrees from the way you'd expect it. So if you want to compare that, you can look at the fructose Haworth projections I showed you earlier on a few slides ago, and you can compare it to what I've done here. And I've just taken it and I've rotated 180 degrees, flipped it around so that anomeric carbon is no longer on the right, but it's on the left. So that was a little tricky. So you get that wrong, don't worry about it, but it's to underscore the rule of how do you find the anomeric carbon? It's the one with 2 bonds of oxygen, and how do you know if it's alpha or beta? You compare it, the highest numbered carbon with that oxygen on the anomeric carbon. Okay, so that's sucrose. Now this is what normally would be called sugar. So if you go in a restaurant and a recipe, you're making a recipe and ask for sugar, they're asking you to put this thing in there. So sugar in common usage is actually sucrose, but when you see sugars on nutrition labels, they generally refer to mono and disaccharides. Okay, so on a nutrition label it says sugars or sugar. They're talking about glucose, fructose, lactose, sucrose, mannose, these mono and disaccharides. Okay, so just so you're aware, that's the meaning. Anything larger than that is not called a sugar on nutrition labels. Okay. So that's joining 2 sugars together, and then you can imagine you can add to that and join 3 or more together. And so then we get polysaccharides, where you get a whole bunch of monosaccharides joined together. And one main reason these are formed is as a form of energy storage, right? And you're probably aware that we use glucose and other sugars for energy, and we'll talk about that when we get to topic 9. But you eat a donut or something and your body breaks that down, you can use that sugar for immediate energy. But if you're eating a lot of sugar, you're going to have more sugar than your body needs, and it's not good to have glucose going around your bloodstream, right? That's and we'll talk about that later too. So we want to store some of that away for later, and the way that you and I do that and other animals do that is in the form of glycogen. And this diagram is trying to show the structure of glycogen. And there's this protein labeled G here. This is called glycogenin. It's just a protein. It's a small protein that forms the core, the basis, the starting point for a glycogen polymer. And then individual glucose monomers are added in sequence to the glycogenin in this sort of structure here. So if I've blown it up on the right-hand side, each one of these little circles is supposed to represent a glucose monomer, and they're joined together in alpha 1,4 linkages for the most part. So then you get a chain of about 13, varies a bit, 12 to 14 glucoses joined together in alpha 1,4 linkages. And then each one of those has 2 points at which an alpha 1,6 branch comes off of it. And this is a repeating structure, right? For glycogenin, you get the first one, and it has 2 branches coming off of it. And then each one of those has 2 branches coming off of it. And then each one of those branches has 2 branches, and so on and so on. So it quickly becomes then this very complicated sort of tree-like structure with all these, fairly short glucose stretches of about 13 joined together in alpha-1,6 linkages. And this, they've shown sort of what you might call 5 shells of these here. Glycogen grows till you get about a maximum of about 12. Okay, so you can imagine this happening, 7 more times, 7 more layers. And that's a pretty big polymer, right? You end up getting thousands of glucoses all joined in one large molecule. Okay, and so just in case there's fuzziness on what we mean by alpha-1,6 and alpha-1,4 linkages, that's what I'm trying to show here. So these are, this along the bottom would be one stretch of 12 to 14 glucoses. And it's carbon 1 of one glucose joined to carbon 4 of another, and an alpha linkage. The O is on the opposite side, it's the highest numbered carbon. And then at those branch points, you get carbon 6 of this glucose joined to carbon 1 of another glucose, and that starts forming another one of those chains of 12 to 14. So this glucose down here in the middle here is joined to 3 other neighbors, right? It's got one at carbon 4, got one at carbon 1, and one at carbon 6, and all the others are joined to 2, sort of hand in hand, 1, 4, 1, 4. Okay? And that's the structure of glycogen, and that's what animals such as you and me use when we have excess sugar. We store them away predominantly in the liver, but also in the muscle. And again, I'll start talking about that and how we use glycogen when we get to topic 9. So that's just planting the seed there. Okay, so that's glycogen. Plants do a similar sort of thing, but a little different. And we call this starch. And the structure has similarities and has differences. There's 2 main components to starch. Amylose, which is essentially alpha-1,4 polyglucose, just a whole big long chain, linear chain. You do find a few branch points, but not very many. So in essence, it's basically a big long chain of alpha-1,4 polyglucose. But most of starch is this amylopectin, which also is alpha-1,4 polyglucose, but has some branches. And it's a little less organized than the animal ones, but about every 24 to 30 glucose monomers, you'll have a branch point. Okay, so it's not as branched as glycogen, but it's the same type of branch. It's the same sort of thing. And plants store these. This is the same. The plants make starch for the same reason that we make glycogen. It's to take excess sugar and store it away in a form that could be accessed later. And because the linkages are the same, like obviously we're able to, when we need glucose, we're able to degrade glycogen to make individual glucose monomers again. And we have the enzymes then to break those alpha-1,4 and alpha-1,6 linkages. And so we can very easily digest starch. So it's, potatoes are high in starch, for example. That's why French fries are good to eat, because one, they may taste good, but also we can access the starch that's in those potatoes for energy. All right. So that's energy storage, polysaccharides. Now, polysaccharides can also be used for structural purposes. And the classic example here is cellulose, which is the major component of woody plants, like wood in trees. And that is polyglucose, but it's got this key difference that it's beta-1,4 polyglucose, and it's essentially unbranched. So this is very much like amylose, with the key exception that the anomeric carbon is in the beta configuration instead of the alpha. And it makes quite a huge difference, right? Think of a potato and how you can boil that and make it all mushy versus a tree. And no matter how much you boil that tree, you're probably not going to get it too mushy because the wood has a completely different structure in 3 dimensions. And furthermore, because it's the beta-1,4 linkage, we don't have an enzyme that will cleave that beta-1,4 linkage in polyglucose. So we don't tend to eat trees for nutrition, all right? Because we are unable to digest the wood. Now, some animals can, or some organisms can, like termites or whatever. They can have the enzymes to do this, but we don't. So cellulose is one example. Now, another example of a structural molecule would be pectin. And I'm showing the structure of pectin here. And it is an alpha-1,4 polymer. And it's mainly composed of this thing called D-galacturonic acid. Now, this is similar to galactose, but not quite the same, because you can see the carbon-6 has a carboxylic acid group on it, whereas normally in a carbohydrate, that would be CH2OH. So strictly speaking, the chemical formula for this is not equivalent to a carbohydrate. However, you can see that structurally it looks very similar to a carbohydrate, and so we would still classify this as a carbohydrate, even though it doesn't meet that formal, CH2O times N formula, okay? But it's clearly very related to a carbohydrate. And sometimes in pectin, you get a methylation of this carboxylic acid. I'm showing that on this one here. Okay, so the chemical formula deviates a bit, but that's okay. We still call it a carbohydrate. But that difference, making it the C double bond or the carboxylic acid, we don't digest pectin very well either. Okay, so our enzymes, as Dr. Botha said, are pretty specific. And so sometimes deviations will make a molecule not a substrate. And there's other examples, lots of other examples of fibers. These are called dietary fibers, right? And by definition, then, dietary fiber is some polysaccharide that we don't digest very well. And so that can just sort of flow right through us, right? We eat the food rich in fiber and our body does its digestive thing and then it comes out the other end. But a lot of that fiber will make it through without being changed too much. Some of that fiber can be attacked by microorganisms that live inside our digestive tract. And sometimes they can use it for energy and convert it to other things. But we would still call that fiber because our organism ourselves, we cannot digest it very well. And there's lots of examples, lots of variations on this theme of carbohydrates. You can imagine different groups on here. Chitin, for example, is one example. That's the exoskeleton of insects, right? It is also a carbohydrate-based molecule. It's got different modifications on it. So the point, again, is not to memorize different structures of fibers, but just to get the idea of the types of structures that fibers adopt, or types of molecules that are considered dietary fiber. And fiber is, been shown to be good for you, for your diet. It's a very maybe complex physiological thing, but it is important to know about your fiber intake so you can remain healthy as well. So if we go back to this nutrition label, now we can see the carbohydrates is labeled. We know what I just defined what fiber is just now. And sugar, as I already told you a few minutes ago, that's mono and disaccharides. So what is missing from this whole grain muffin, the 31 grams of carbohydrates that are not listed here, that's likely starch, right? Because the muffin is a plant-based food product and plants make starch. So the carbohydrates there are probably starch, right? And if it's an animal-based thing, you might have some glycogen in there, but they're still not going to put that there. So what's missing here are complex carbohydrates that we can easily digest, like starch or glycogen, okay? But no glycogen here because these are all plant-based foods, okay? And just lastly, another function that I thought I should mention of oligosaccharides, as I've said before, you can find them on the surface of cells. What are they doing there? Well, one thing they do out there is protection, just simply making a zone, like a barrier to protect, cells physically. Just sort of, you can make layers of and networks of carbohydrates, perhaps, that will provide some physical cushioning. Also slippery, and this is because the carbohydrates have a lot of OH groups on them, and that tends to attract water. And so if you've ever picked up like an earthworm or something, or some animal, some organism that's got mucus or stuff on it, that's largely carbohydrate based. And it's slippery because of those carbohydrates. And that helps cells slip past each other, like red blood cells, for example. They've got to slip into very tiny blood vessels. And if they were too sticky, that wouldn't be a good thing. So carbohydrates can help them get past each other. And also some carbohydrates are functional in that they mediate cell-cell recognition. So this is just a figure I grabbed from this old review paper, but these different shapes here are supposed to be showing sort of an oligosaccharide on the surface of this membrane protein here. And there might be receptors on another cell that are, you can see the shape is sort of specifically matched to this carbohydrate. So this can help individual cells recognize other types of cells. And this is really important. If you think back, way, way back, before you can even remember, each one of us started out as a single-celled organism, and that single cell came from the mating of a sperm and an egg. That interaction between sperm and egg is carbohydrate mediated. Okay, that's how those two types of cells recognize each other. So this is obviously important for everybody because this is how we all started out, through this sort of interaction between two cells. Okay, so that's covered some major functions of carbohydrates. They're used for energy storage, structurally, and on the cell surface for different reasons. And that takes us to the end of topic 7. And again, I'm not going to read through these different summary points, but you can reflect on them later and see how we've been there. So I got 10 minutes left, and I am going to start into topic 8. Yeah, I know. I'm cruel that way. Why? Because we got to motor through. We want to cover everything. We look at all the stuff we got to cover yet before the midterm. So we're going to just start on topic 8. Don't worry, I won't tax your brain too much today. Where we're going is the difference between delta G and delta G with the little circle. We're going to talk about different types of reactions, spontaneous or non-spontaneous. How do you drive unfavorable processes forward? And we're going to define some terms in metabolism. We're going to talk about how metabolic pathways are regulated in various ways. And in the end, we're going to talk about these electron carriers, okay? But to start, we're going to talk about Gibbs free energy. Now, again, sometimes this gets people a little on edge because this is not a thermodynamics course, and we're just going to talk about this to the very minimum depth that we need to talk about metabolism, okay? But Gibbs free energy should be a concept you've been exposed to before. And then when you have a reaction like this, you've got some reactants and products, and the reactants have some amount of energy or Gibbs free energy, and the products have some amount of Gibbs free energy. And then we define this reaction by the delta G, which is the Gibbs free energy of the products minus the Gibbs free energy of the reactants. And before I go further, I do want to, show this guy. This is Gibbs. In case you ever wondered, Gibbs free energy, who was Gibbs? Well, here's this photo anyway. And you can see no Nobel Prize for Gibbs. Well, even though Gibbs free energy is pretty, it's taught to every single science undergraduate in the universe, right? So why didn't he win the Nobel Prize? Well, the Nobel Prizes they just started giving them out in 1901, okay? And the other rule they have for Nobel Prizes is they don't give them to dead people, okay? And so you can see that Gibbs here died in 1903. And I have to think that if he just was able to hang on a few more years, he might have won a Nobel Prize, just because this is such a fundamental concept, and the thermodynamic stuff that he sorted out is pretty fundamental. So anyhow, Willard Gibbs, there he is. Fun fact, he never got married, and he lives his entire adult life with his mom. Take that, good or bad, whatever. It's good to love your mom. Anyhow, so Gibbs free energy, here he is looking across, and he's looking and seeing that the delta G is affected by two major things. You can classify it in your brain this way. Like there's properties of the molecules, right? So there's just types of reactions that happen. The molecules are what they are, and they undergo a specific reaction, and you can't really change that. And that's going to influence the delta G. But then there's a layer on top of that, which is the environmental conditions, or the conditions that reaction proceeds under. And that can change the temperature, the pressure, things like that, concentrations of these. And so the delta G is not going to be a constant. It will depend on the environmental conditions, but it's also largely influenced by the type of reaction that's happening. So these are sort of interplay. One thing you can change, but one thing you can't. Okay. Now, it's back again. So we're going to talk about delta G now in different circumstances. So in this case, we can look at the reactants, and we see that the Gibbs free energy is higher than the Gibbs free energy of the products, right? And in this case, the delta G is less than 0. And what that implies is that the products are more energetically stable than the reactants, right? And I've shown it going through some reaction intermediate, and I'm sure Dr. Bofa talked about that, but every reaction pretty much has this intermediate stage. You sort of get a higher energy form, but then that resolves into the products. And so if you come across a situation like this, and you observe it over time, what you will see is net conversion of reactants to products. As time passes, some reactants will be converted to products. And that's called a spontaneous reaction. You don't have to do anything to help this along. If given enough time, the reaction will go from left to right as written, right? And so I've shown this as an equilibrium because every reaction in principle is reversible and that the products can go to reactants, and that's conceivably possible, but it's not as favorable as reactants going to products. So in a net sense, if you consider both directions and add them up, so to speak, you do get net conversion of reactants to products. This is a spontaneous reaction. Now, the Gibbs free energy of the reactants and products depends in part on their concentrations. That's one of the environmental conditions that influences the Gibbs free energy. And it turns out that the higher the concentration, the higher the Gibbs free energy. So if you can imagine this reaction proceeding, and if you give it enough time, what will happen is the reactants will be converted to products, and you can imagine then the concentration of reactants will start going down. And the concentration of reactants or products will start going up. And so you'll eventually reach a state where the Gibbs free energy of the reactants falls and the products rises to the point where they're equal. And this is what we call equilibrium, right? And this should be hopefully review from chemistry. And at this point, the forward and reverse reactions are going at the same rate, and you get no net conversion of reactants to products or products to reactants, right? And that's equilibrium. It's not that there's no reactions happening, it's just the rate of the forward reaction equals the rate of the reverse reaction. And here, the Gibbs free energy equals the delta G equals 0. And then conversely, you can imagine a case where we write a thing we call reactants over here and products over there, but you come across a flask or a test tube and you say, oh, the concentration of products is really high and the reactants is really low. And in fact, the products have a higher Gibbs free energy, and the delta G would be greater than 0, and this would be called an energetically unfavorable or non-spontaneous reaction. And in fact, what will happen is you'll get conversion of products to reactants. So basically, we've labeled the equation in the wrong order. We sort of put the products on the right when we should really be calling them the reactants in this case. But you know, if reactions can go back and forth, it's just a matter of perspective. What are the reactants and what are the products? Okay, but in principle, then you can imagine a case like this where the reaction will go from right to left spontaneously as drawn. And if you're considering the forward direction, that would be unfavorable. So I've drawn that little arrow much smaller. So the forward reaction here, reactants to products, is unfavorable. That's non-spontaneous. In fact, you'll get the reverse reaction happening. Okay, so those are different types of reactions. Now, let's think about Le Chatelier's principle. And again, this hopefully is review for you, but we can imagine a case where we have some a reaction that's reached equilibrium, but then we change the concentration of one of the reactants or the products. So let's imagine we increase slightly the concentration of the reactants. That's going to increase the Gibbs free energy. Well, what's the response of this system going to be? Well, now we have a system where the delta G is negative, right? Slightly negative. There's the higher Gibbs free energy for the reactants and the products. And what will happen is this will spontaneously tend to move back towards equilibrium, right? You'll get net conversion of reactants to products here. And so the reaction, you could say, will shift to counteract the change in concentration. We increase some reactants. Some of those reactants will be converted to products. And again, if you leave that go long enough without disturbing it again, the reaction will return to equilibrium. It's just you'll have a bit higher concentration of both reactants and products in the system at that point. Okay? And the same thing happens in the other direction, right? And yeah, we're back to equilibrium. This reaction is said to be reversible in that, like biochemically, this is what we'd call it. The reaction is poised sort of at an equilibrium point where, depending on the relative concentrations of reactants and products, it could go in one direction at one moment, and then a few minutes later, it could be going in the other direction. It's sort of like a poised at this balance between the two, and we call that reversible. So that's just sort of a way biochemists talk about it. And this is Le Chatelier's principle. Basically, any change you make to the system will sort of counteract that to try to go back to equilibrium. And here's Henri Le Chatelier, Nobel Prize for him. And it occurred to me when I found this picture of Le Chatelier that we often see these photos of these old guys when they're old, because, when he was young, like, who'd want to take his photo? He hadn't figured out Le Chatelier's principle yet. He wasn't famous. But I did dig a bit and find a picture of Henri Le Chatelier as a younger man. And here he is in some military uniform. And I like this because he is wearing a sword. And I think that would make scientific debates a lot more interesting if we all had swords and we could really start to duel if we disagreed about stuff. Probably maybe not the best way to solve your differences, but something to think about just the same. Okay, so we will see you Friday, or no, Thursday. This group is coming Thursday. See you tomorrow.